Hello everyone, welcome to our session on sex determination. So in the last session, we were discussing about the different types of sex determination, which is XX, XY, as well as ZZZW. And in this session, we are going to discuss about the sex determination in honeybees. So in a honeybee colony, there are three types of individuals: the queen, the workers, and the drones. Of these, the drones are the only males which are developed by the particular process which is called as parthenogenesis. So, parthenogenesis is nothing but the development of an egg without fertilization. An unfertilized egg, or the direct development of an unfertilized egg, is usually referred to as uh, parthenogenesis. And because it is because these individuals are developed from an unfertilized egg which is resulted after an oogenesis, the process of meiosis after reduction division. And for diploid organisms, the egg will be haploid. And such a haploid egg without fertilization is developing into an organism, and that organism will also be haploid. So the sex determination methods of drones is referred to as. So this particular method of sex determination is referred to as male haploidy. That means the males will be haploid, or haplodiploidy, or arenotoky. So all these refers to the same process of development of drones in the honeybee colony. That is the development of an organism without fertilization or from a haploid egg. And the resulting individuals, all the cells of the resulting individuals will also be haploid. The queen chooses the eggs not to be fertilized by the sperm, which is stored in the seminal receptacle, which will develop into males, and hence they are haploid. And what happens in this haploid drones is that, so haploid drones are the only males in the colony, and they are the ones which stores the sperms in the seminal receptacle of the queen. And in such a process, what happens is, in their body also, the process of gametogenesis should happen for the process of the production of sperm, which is called as a spermatogenesis. So during the process of spermatogenesis, the usual process of meiosis will not happen, because the usual process of meiosis is a reduction division. And in a drone which is haploid, if reduction division happens, the chromosome number will go again half. As a result, when fertilization need to occur, from or a proper haploid gamete will not be formed in the case of drones if meiosis happens. In such a cases, they are having a special type of meiosis where there is no recombination, there is no crossing over, and there is no reduction division. So such a division is more or less similar to what is called as a mitosis. So they have a particular process of gametogenesis, or without reduction division and crossing over, and the sperms produced will be haploid. So that is, the sperms are produced by a process which is similar to mitosis. So their body cells are all haploid. As a result, the sperms produced also is haploid, and that is because of a process which is similar to mitosis, without reduction division, without crossing over, and without reduction division. So this is how this is what a drone in a honeybee colony means, and this is how sex is determined in the drone. So here, sex determination in the drone is not completely genetic. So it can be called, or it is categorized under this chromosomal theory, because all the haploid, because haploid in the sense, if the queen is not fertilizing the egg, they will develop into males. So the genetic part of the sex determination in such the development of a drone is that they are haploid. So haploid eggs are developing into, or haploid eggs without fertilization are developing into the drones. So which egg is to develop into, or every egg has a potential to develop into a drone, because if that egg is not being fertilized, it will develop into a drone. So here, the determining factor is completely not a genetic in nature. It is the discretion of the queen. So here, the queen is the determining factor. The queen decides who should be male and who should be female worker. So here the main determining factor in the development of the drone as well as the worker. In any case, it is the discretion of the queen. So this is how drones are produced in in a honeybee colony. Coming to the queens and workers, queens and workers both are diploid females. And the difference between the queen and the worker is that the queen is the only fertile female in a honeybee colony, and the workers are females, diploid females, and they are sterile. So they do all the housekeeping duties inside the colony, and they don't have a chance, or they do not have any chance uh, to be forming a fertile female. So there will be only one fertile female in the honeybee colony, and that will be the queen. And what decides? So the genetic constitution of the queen and the uh, worker is the same in a honeybee colony. Both of them are diploid. The only difference, diploid females, and the only difference is that one group is sterile and the other one is fertile. And this depends on the amount and the quality of the food that is made available to the diploid larva, and determines whether the female becomes a sterile worker or a fertile queen. If that particular this happens, or the female or a worker is developed, or it is being developed when the egg is getting fertilized. So the previous queen in the honeybee colony, if it is fertilizing the egg, that they will develop into all will be diploid females, because a haploid sperm and a haploid egg, they fertilize to produce what is called as a diploid uh, a diploid female. And that diploid female, one of the diploid female larva will be fed by quality food and also the more amount of food, and that will develop into a fertile queen. So here the environment determines sterility or fertility, uh, but does not alter the genetically determined sex. So in this case, the genetically determined sex is not altered. The same is the case with drones. Their genetically determined sex is not getting altered, but the deciding factor is the queen. So here the environment in the sense, what is meant by environment in the sense, it is the quality and quantity of the food, what is mentioned or what is referred to as environment in this case, determines sterility or fertility, but does not alter the genetically determined sex. The sex ratio of the offspring is under the control of the queen. That is usually there will be only one a honeybee or a queen in a honeybee colony, and it will meet only once. And how it is maintaining the sex ratio in that, uh, the queen is the one decides. If there are 100 eggs, it is the queen that is deciding how many will be fertilized and how many will not be fertilized, and that will decide the ratio, how many males and how many females. That is why we say that the sex ratio in this case is under the control of queen. So this is this, type of sex determination that is seen in honeybees. Next is the second type. So we have come to the end of the first type, which is called as the chromosomal theory of sex determination. Coming to the second one, which is the genic balance theory. And this particular genic balance theory is explained on the basis of the studies in Drosophila by Bridges. The critical factor in the case of Drosophila in determining the sex is the ratio of X chromosome to the haploid set of autosomes. So, to make it more clear, we have the formula which is given here. So, here in the case of Drosophila, Bridges proposed that the sex in Drosophila is not determined by whether they're having. We said that they're having an X chromosome. They are having something which is called as XXXY system. We discussed about that in the earlier session. But here Bridges proposed that even though they are having the XXXY system, the determining factor whether that fly to be male or that fly to be female is determined by this particular ratio, and that is the number of X chromosomes. So here the Y chromosome does not have any role, and the number of how many X chromosomes are there in that particular individual divided by the uh, number of sets of autosomes in a haploid set. It is not a complete autosomes. It is the X chromosome to the haploid set of autosomes. Haploid set means if you have four, it will be uh, it will be denoted by, or it will be 2A. Okay, so that is how you determine this particular ratio or this particular haploid set. Here it should be given as haploid set of autosomes. So please don't take this as a mistake. So it is the number of set of autosomes, and the set of autosomes is haploid set, not the complete set of autosomes. And that is a ratio, and that is the ratio that determines whether a fly is a Drosophila is a male or a female. And the reason why we do not consider Y chromosome, because they are having an XXXY system, the Y chromosome should be considered as a determining factor to determine whether the fly is a male or a female. So here in this particular genic balance theory, it is proposed that in Drosophila, the X chromosome carry the female determining genes, but the male determining genes are present in the autosomes and not in the Y chromosome. In the case of Drosophila, Y chromosome does not have any part in determining the sex of the male, and it is usually said to be genetically blank with sex or sexually neutral because they do not have any genes that determine the sex of the individual. So, sex in Drosophila is determined by the number of chromosomes divided by the haploid number of autosomes in the or haploid number of autosomes. So, likewise, Bridges from his various experiments and also by observing many Drosophillas, he arrived at different types of individuals depending upon this ratio without considering Y chromosome. Only without considering this ratio, you can find out the sex of the particular Drosophila. And what he proposes that by if this ratio is equal to 1, so when this ratio should be equal to 1 means there will be the number of X chromosome and the number of haploid autosomes. So in Drosophila, you have 4 chromosomes. We discussed about that in the last session. They are having uh, 4 pairs of chromosomes, okay, of which 4 pairs of chromosomes are there, and that what happens is that in that three are autosomes, or 3 pairs are autosomes, and 1 pair is the sex chromosome. So here, a female is produced when you have a haploid set of autosomes, and which is divided by the number of X chromosome divided by the haploid set. So in these two cases, so here you can see you have 2 X chromosome, and in this is a haploid number. If the haploid number is 2, and here the haploid number is 3, even though there are more X chromosomes, you can see that the sex ratio will be, or this particular ratio, the sex index ratio is 1, that is 3 by 3, this is 2 by 2. The ratio will be 1, and that will be a normal fertile female. So even though this is the X chromosome is more here, considering the sex index, this if it is 1, this is a normal fertile female. Coming to the next ratio, if the sex ratio or the sex index is 0.5, that will be a normal, that will be a male. And to be normal, it should be having an XY, which is a fertile male, because in both these cases, these are the normal ones. That is a haploid pair of autosomes here, and these are the haploid pair of autosomes, and this one is the sex chromosome. If it is X and Y, the number of X chromosome is 1, and you have 2 autosomes here. That haploid number is 2, that is the half of 4. So what happens here is 1 by that is 1 by 2 you have, which is 0.5. Here also the sex ratio is 1 by 2. So even though the sex index is 1 by 2, because we discuss if this is 0, it will be a sterile male. Okay, and coming to the next ratio, if the ratio is 1.5, that will be a super female or a meta female, and that is sterile. So meta female is occurs when you have this particular, that is 2A autosomes haploid set, and 3x, that will be 3 by, or you have 3 by 2, which is 1.5, and that will result in, if this index is 1.5, you have a sterile super female or a meta female. The same is the case if it is less than zero point 0.5. Less than zero point 0.5 is you have more number of haploid set and lesser number of X chromosome. This will be 1 by 3, and 1 by 3 will be less than zero point 0.5, and that will result in a super male or a meta male, and that will be sterile. And if this particular ratio is between zero point 0.5 and 1, it will result in a condition which is called as an intersex, which is also sterile. So 3A and double X. So here also it will be 2 by 3, which will be between zero point 0.5 and 1, and that will be an intersex. So what Bridges does is, so he observed this normal cases, that is the female and this normal male in normal Drosophila groups, and these were generated by different process. So he crossed different Drosophila to find out how this particular flies will be, and these flies they developed they showed this particular type of autosomes as well, haploid autosomes as well as X chromosome, and they were sterile. So these were experimentally derived by Bridges to know whether how these sex ratios affect this the number of X chromosome is affecting their genetically determined sex. So from this we conclude that in Drosophila it is a genic balance theory that determines whether, apart from whether the presence or absence of Y chromosome, the sex of the particular individual is determined by the genic balance theory. That means it is the ratio of what is called as a sex index, which is the number of X chromosome divided by the haploid number of autosomes. So this is what is called as genic balance theory. So coming to intersexes, what we discussed last in the case with Drosophila, intersexes and Gynandromofs. So individuals having an intermediate sex characters between males and females, and they are mostly sterile, and the reproductive organs are intermediate between testis and ovary. And British produced them by crossing triploid female with a diploid male. And the genotypes are 3AA plus XX or 3AA plus XXY. Here the sex index is 0.67. So that is between 0.5 and 1, and that will result in an intersex. So intersex is having an intermediate sex characters between males as well as females, and they are sterile. And here you can see that the reproductive organs are intermediate. So it will be a crippled type of reproductive organ. Some parts will be as that of a male one, and the other part will be that of a female one. So it will be also an intermediate one, and the sex ratio is 0.67. So the next one is Gynandromorphs. In the case of Gynandromorphs, Gynandromorphs were also discovered by Morgan and Bridges. This is also a special type of or a special group of flies. So here the flies, the speciality is that the flies are one half of the body will be male and the other half will be female, and they are called as Gynandromorphs. That is, one half male and the other half of the body will have one half of the body will be having male type of cells, and the other half will be having a female type of cells. So they can be called as sex mosaics, and they are usually sterile. Like intersexes, they are also sterile. So they contain cells in their body which are genetically males and genetically females, and the amount, that means how many cells should be male and how many cells uh, are female, vary in different types of gynandromorphs. And accordingly, the three types of gynandromorphs are distinguished. They are bilateral gynandromorphs, anterior posterior gynandromorphs, and also sex p-balls. So in bilateral gynandromorphs, what happens is that if I consider this as a Drosophila fly, so the shape is slightly crippled. So if this is a Drosophila, bilateral gynandromorph means if I cut this organism bilaterally, one half will be male and male cells, and the other half will be having female type of cells. It can be also opposite, the right and the left. So whether if this side is male, this side will be female type, and this side is male, this side will be female type. So vice versa can happen. Um so such a type of gynandromorph is called as bilateral gynandromorph. So coming to anterior posterior gynandromorph, the same thing, anterior posterior. So in anterior posterior, if this is the organism, so this will be the anterior one. So if this is male, the opposite, the posterior side will be female. If anterior is female, posterior portion will be male. So this is anterior posterior. And next is sex p-ball. So sex p-ball is entirely different from what we discussed, the anterior posterior and bilateral. uh, Here, if this is the organism in sex p-ball, if I take this particular one as female, so the entire portion is female, you can see some clusters like or islands or group of cells in the body. Okay, in some clusters you can see, and these clusters will be the opposite sex. So if the entire body is female, the clusters or the islands which you see here will be male. If the body is male, the clusters that you see here will be female. So the body of one sex has scattered spots of the other sex or the other sex tissue, and that is what is called as sex p-balls. So now we have to know whether what is the difference between intersex and gynandus. So we said that both are sex mosaics, both are having both male and female characters. So what is the actual difference between an intersex and a gynandromorphs? So both are, as we said, both are sex mosaics, and intersexes are the cells, or intersexes have the cells which are genetically alike. Genetically alike in the sense they are having the cells which are neither male or female. Okay, neither male or female cells. That means all the cells in their body are same. All the cells in their body is called as intersex. Okay, you cannot distinguish one cell in its body to be male and the other cell to be female. In an intersex Drosophila, all the cells on the body is the same, and that is having the sex ratio as we discussed earlier. This is having all the body cells is having a sex ratio of this. So it can be either in this form or it can be either in this form. So in any case, the sex ratio is this particular one, and if this happens, all the body cells will be intersex. Okay, you cannot distinguish a male cell or a female cell in an intersex Drosophila. But when you come to Gynandus, as we discussed now, the in Gynandus you can clearly distinguish the male cells as well as female cells separately. That is, some cells are genetically males and the other cells are genetically male. So this is about the different, that is unusual type of sexes in Drosophila, and that comes along with our genic balance theory, because in genic balance theory we discussed everything like super female, then the super male, then intersex, etc. So we discussed some of the unusual sexes which is seen in Drosophila, like the intersexes as well as Gynandus or Gynandromorphs. In Gynandromorphs we discussed three different types. One was bilateral, anterior, posterior, and sex p-balls. And we also discussed what is the difference between an intersex and a gynander. So the main difference is that in intersex, all the body cells are intersex cells. Okay, you cannot distinguish a cell if you take a particular cell from an intersex Drosophila, and if you observe it, you cannot say that whether it is a male or a female. The cell can be called as an intersex with a sex ratio 0.67. But when you come to gynandus, if you take one cell from the body, it will be male. If you take a cell from another part of the body, it will be a female cell, a normal female cell, without any unusual type of this particular any unusual type of chromosome. It will be a normal female cell and a normal male cell, but both are present in the same individual. That's why they are called as gynandus. So how this, or what is the reason why you get gynandrome? So there are two theories which are proposed. So one is seen in Drosophila, and the other one is seen in other insects. The second one is usually seen in other forms of insects. This one is proposed for Drosophila, that is the elimination of X chromosome. So a fertilized egg usually have two X chromosome. That means fertilized egg, it means a zygote. It is having two X chromosome. And during cleavage, that means after fertilization, you get a zygote. The zygote starts to cleave. So one cell divides into two, two divides to form four cells. So in such a stage, what happens is one of the blastomere. So at early stages, you cannot say whether it is a two cell stage or four cell stage. So in the early stages, the development has not proceeded much further. In the early stages, one of the blastomere, a blastomere is nothing but one cell of a zygote. So after cleavage, the fertilized egg, the zygote, it divides into two. So that two cells are called as blastomeres. That two blastomeres divides into four, and it goes on. So of that, one of the blastomere will lose one chromosome. As a result, the cell arising from this particular blastomere will have only one chromosome. That means one of the blastomere. So I consider I'm having now, for say, two blastomeres are there. Okay, this is a normal blastomere with a normal X chromosome. So when this divides, it will give rise to, again, because it is mitosis and equational, it will give rise to two cells which is having two X, etc. But what happens to this particular blastomere is that this particular blastomere has lost one of the X chromosome. As a result, the cells which is arising from the division of this blastomere, so both this is contributing to the body of the same Drosophila. So the cells arising from this will have only one X chromosome each. There will be a lacking of another X chromosome, that is X0. Okay, and the other cell will be normal, giving rise to two X chromosome. So here the cells from with only one X chromosome will be the male cells. They are sterile males. X0 will be male cells, and the other will be two X chromosomes, that will be female cells. So, in one organism itself, you are having two types of chromosome. One group of cells with two X chromosome, the other group of cells with one X chromosome, and that one X chromosome X0 will be a sterile male cell, and the other will be a normal female cell. So, this is one method by which a gynandromorph is generated in the case with Drosophila. Coming to the other one, fertilization with a binucleated egg. This usually happens in other group of insects. So during oogenesis, here what happens is, uh, in reduction division and in oogenesis, a small after meiosis 1 and meiosis 2, the cell becomes 2, and that smaller cell is called as a polar body. And in such cases, the polar body is retained inside the oocyte, forming a binucleated egg. It is retained inside the oocyte and it forms a binucleated egg. So the egg now contains two nuclei. Actually, the polar body is the one which is to be released outside, but retaining that, now the egg is having two nuclei, one with X chromosome and the other with Y chromosome. So these are the insects which are having the opposite type, that is XY or ZW type of uh, female, and ZZW male. So during fertilization, if two sperms are fertilizing the two nuclei, it will give rise to a male as well as female tissues. Okay, that is, one nucleus will be male and the other nucleus will be female. And during development, what happens is you will have an insect with two different type of cells in a particular insect. So this is the mechanisms which are proposed by how gynandromorphs are generated, or how gynandromorphs are developed. So the next is another thing which is also an unusual type of sex, which is called as hermaphroditism. So organisms with both the ovary and testis is called as hermaphrodite, and the phenomenon is called as hermaphroditism. So in man, hermaphrodites are usually having an ambiguous external genitalia. Ambiguous in the sense that their external genitalia in males should have a penis as well as a scrotum with two testes, and the other thing, in females they should have the structures, the opening which is the openings of the vagina, and also the other associated structures, the clitoris, etc. All these are considered to be the external genitalia, coming to the case with females. In humans, so what happens is, when you have a hermaphrodite in the case, in humans, they will have ambiguous in the sense the sex organs will be a mixed type. So, if a penis is formed, it will not be formed completely as a normal penis. It will have some deformities, and some cases the penis will be present, and also the female structures or the female openings will be present instead of scrotum and testis, etc. So, it can happen depending upon the errors in the development which is happening in a hermaphrodite. Such an unusual, that is why you call it as ambiguous, unusual type of external genitalia can be seen. So hermaphrodites are generally sterile, but one case is reported in 78, and it is that a 25-year-old hermaphrodite delivered a stillborn child after 30 weeks of gestation. So it was stillborn; it was not a fertile young one is not produced in that case. So this is hermaphroditism in the case with humans. Uh, so they are having ambiguous external genitalia, which we cannot determine whether it is a male type or a female type. So next is and the other topic, which is called as Barbody and Lion hypothesis, which will be discussed in the next session. Thanks for hearing.